Okay, our next speaker is Jessica Nelson from the Granado Lab at UPenn, and she will be talking about regulation of habituation learning.
So my work focuses on learning. Learning is something that I'm guessing you guys are all pretty passionate about, and that's why you're still here at 9 o'clock. For the purposes of this talk, I'm going to define learning as the process that allows animals to modify their behaviors by incorporating new information. And I'm going to be talking about one specific example of learning called habituation. So habituation is the process by which animals respond less to a non-threatening stimulus with repeated exposure. And so on the left, this is a graph of habituation. We have increasing trial number, and the animal is responding less as they're exposed more to the same stimulus. And habituation is learning in its simplest form. So it's very easy to study in the lab. We can study habituation to a lot of different stimuli, but the one that we've selected in the Granado lab is the acoustic startle response. Zebrafish larvae exhibit stereotyped acoustic startle responses at 5 days post-fertilization, and we can track them automatically. So this is a video of the acoustic startle response in the 5-day-old zebrafish, and you can see that it's going to make a unilateral body contraction and then execute a swimming bout. We can visualize these acoustic startle responses in a very high throughput fashion by arraying zebrafish larvae on a petri dish. They each get their own well, and then we attach that petri dish to a vibrational exciter, which delivers acoustic stimuli, and then we record their movements with a high-speed camera. We can then track those movements using some software that was developed by a former postdoc in the lab, Harry Burgess, and analyze them very easily. And by analyzing tons and tons of fish over many, many years, we've determined that the zebrafish acoustic startle response is subject to habituation learning. So this graph should look very familiar to you. Looks just like the one from my first slide. You can see that if we present acoustic stimuli once every 20 seconds, zebrafish larvae startle almost every single time. So that's here. And then if we shorten that interstimulus interval to 1 second, the zebrafish larvae will very quickly learn that the acoustic stimuli are not threatening, and they'll stop responding. Here. And it's worth mentioning that habituation in the larval zebrafish fulfills the 9 behavioral characteristics that were established by Thompson and Spencer in 1966. So I'm telling you about some really simple behaviors, right? Acoustic startle response and habituation of the acoustic startle response. But thresholding of the startle response is actually dysregulated in a number of human neuropsychiatric disorders, including these really, really common ones. So anxiety disorders, schizophrenia, autism spectrum disorder. And so we think that this question of understanding learning is not only interesting from a basic science perspective, but also from a more clinical perspective. So I've told you so far that habituation is a tractable form of learning. And although habituation is disrupted in these human diseases, the genes that are controlling habituation are not known. And so this has been our goal in the Granado lab. So how can we get from genes to this behavior that we're interested in, specifically habituation? Well, we took a forward genetic approach, and so a couple of postdocs in the Granado lab took several years to perform a forward genetic screen, and they identified a bunch of genes that are required for habituation. And this has been a really, really successful screen. So this is just a list of the mutants that we've mapped. So there's even more than this. And in pink are the mutants that have been mapped that have habituation phenotypes. But I'm not going to tell you about any of those today. I'm going to tell you about my mutant. So my mutant is called slow learner. And slow learner mutants, hence the name, cannot habituate to acoustic stimuli. So first I want to say that these fish have pretty normal acoustic startle performance. So if I play a loud acoustic stimulus, the wild type fish and the slow learner fish will both execute those sea bends and the swimming bouts. But if I shorten the interstimulus interval and try to habituate these fish, you'll see that although the wild type fish is pretty relaxed, chilling out in the corner, he doesn't find this threatening at all, the slow learner fish still finds these acoustic stimuli terrifying at stimulus number 17, and even stimulus number 35. Slow learner just doesn't get it, so hence the name. Okay, so this is just a quantification of that phenotype. So whereas the siblings very rapidly habituate to acoustic stimuli, slow learner mutants never habituate during my protocol. And this is in spite of the fact that slow learner mutants have normal body morphology, except that maybe you guys noticed they don't have a swim bladder. They have normal acoustic startle performance, and they have a normal touch response. So of course, we want to know what is the molecular nature of the slow learner mutation. And so to get at this, we undertook whole genome sequencing. And when I subjected these fish to whole genome sequencing, I identified that slow learner mutants have an early stop codon in a gene called HIP14. HIP14 is a palmitoyl acyltransferase. It palmitoylates its targets using this DHHC domain, which is characteristic of all palmitoyl transferases. But HIP14 also has these unique N-terminal repeats, which it uses to recognize its targets. And HIP14 is named for its most famous target, Huntingtin. So HIP14 stands for Huntingtin-interacting protein number 14. And you guys have probably heard of Huntingtin because this is the gene that causes Huntington's disease, a very devastating neurological disorder. And again, HIP14 interacts with Huntingtin, as you can see here, via its N-terminal anchorin repeats. So my slow learner mutants have a premature stop in HIP14 at amino acid number 212 out of 620. This truncates the protein, and so you can see here that it eliminates all of the conserved domains, including 2 anchorin repeats, all the transmembrane domains, and the DHHC. And we think this is a loss of function because I have generated CRISPR mutants that cause frame shifts and early stops that also recapitulate the phenotype. And so next I was really interested in these N-terminal anchorin repeats. And again, that's because these are not typical of palmitoyl acyltransferases. And so we wondered whether this Huntingtin-interacting motif would be important for HIP14's role in habituation. And so in order to determine whether HIP14 requires these highly conserved N-terminal sequences for its function, I used my CRISPRs to generate in-frame deletions. And so I recovered an in-frame deletion of amino acid number 78. This should result in an intact protein that just has a deletion of a single amino acid. And much to my surprise, this resulted in a pretty severe learning deficit. So whereas the HIP14 siblings rapidly habituate, and you can see my control up here, the HIP14 slow learner mutation that came out of our INU screen, failing to habituate. And you can see that the deletion of only a single amino acid also has a very severe learning deficit. And so although these are preliminary findings, you can see I've got an N of 4 right there. But we think that this suggests that the Huntingtin-interacting interface is required for HIP14's function in habituation. And so next, we wanted to know where is HIP14 acting within the habituation circuit. And lucky for us, the habituation circuit is pretty well described in larval zebrafish. So these hindbrain neurons called the Mauthners are necessary and sufficient for the acoustic startle response. They receive their inputs from the 8th nerve at the lateral dendrites. And so we had a couple of hypotheses as to where HIP14 could be, for how the Mauthner could be defective in these HIP14 mutants. One is that the Mauthner is hyperexcitable. So in this scenario, inputs to the lateral dendrite are diminished, but somehow they're still sufficient to drive firing of the Mauthner neuron. In the second scenario, the defect is in the lateral dendrite or upstream of the Mauthner. And I'm going to show you through some calcium imaging data that it's actually more likely to be the second of those two options. So in order to answer this question, we used calcium imaging. I used a setup that Kurt Marsden, a postdoc in our lab, had developed, where I head fix larvae in a block of agarose, leaving the tail free to deflect back and forth as a readout of the C-bend behavior. And you can see that I can put those fish in the block of agarose onto a confocal microscope, visualize activity in the Mauthner, simultaneous with the tail deflection behaviors. And very much to my surprise, when you head fix a fish that's mutant for HIP14 and agarose, this does not improve their learning behaviors. So even on that calcium imaging setup, they fail to habituate. Siblings habituate very well. And not surprisingly, the Mauthner soma seems to be tracking that behavior. So whereas the siblings very quickly stop firing their Mauthners, the mutants continue to fire throughout the habituation protocol. But then at the lateral dendrite, although the signals in the mutants start out a little bit lower, I hope you can appreciate that the slope of these two lines is very different. So whereas the signals in the lateral dendrite diminish very rapidly in the siblings, you can see that in the mutants, they have much less diminishment in their signals in the lateral dendrite during habituation. And so we think that this suggests that the HIP14 defect occurs in the lateral dendrite or upstream of the Mauthner. So I've told you today that HIP14 is a novel regulator of habituation learning. I've told you that the requirement of the HIP14 anchorin domain suggests a novel role for Huntingtin in habituation learning. I've showed you some calcium imaging data that suggests that HIP14 acts at or upstream of the Mauthner lateral dendrite to mediate learning. And moving forward, I'm looking forward to testing the role of Huntingtin in habituation learning and identifying the cell types in which HIP14 acts to mediate learning. And in order to do that, I have a couple of really good candidates, since this is a fairly well-described circuit, but I plan to take an unbiased approach, looking at immediate early genes to try and identify cells that are differentially active in my mutants versus my wild-type fish during habituation learning. So I'd like to thank my lab, especially my mentor, Mikel Granado, the 3 postdocs who undertook this awesome forward genetic screen, Mark, Roshan, and Kurt, along with a couple of technicians, Hannah Bell and Julianne Skinner. I also want to thank the fish facility and Katharina Hare, who helped me to analyze my whole genome sequence data and my NRSA funding. And check out Hannah Schoenhard's poster and Catherine Gribel's talk and Patty Murphy's talk as well on Saturday. Thank you.
I may have missed this, but is HIP14 novel, or what is known about it in other animals like mice and so on?
Yeah, so HIP14 hasn't previously been shown to have a role in habituation learning. It was identified in a yeast-2 hybrid screen for Huntingtin-interacting proteins, and so this is a novel finding for HIP14 to be involved in learning. Yeah. Thank you.